Hi, everyone. My name is Bruna, and I am a graduate student at the University of Melbourne. Today, I will talk about predicting conformational B-cell epitopes using graph-based signatures. My supervisors are Dr. David Asher and Dr. Douglas Pires. And in this presentation, I aim to answer four main questions. What is B-cell epitopes? Why is it a relevant topic? How our tool predicts these epitopes, and what is actually our contribution to this area? And to answer the first and second question, what is and why epitope is a meaningful topic, it's important to remember a biological concept here. B cells are an essential part of the immune system that provide long-term protection against pathogens because of antibodies. So how antibodies identify an antigen and eliminates it from our body? In short, they bind to them. They connect to a fragment of the antigen called epitope. So epitopes are classified in two types, the conformational and the linears. But most B-cell epitopes are conformation, around 90% of them. And they are also called discontinuous, which has made their identification, the discontinuous, so tricky, as they are often composed of residues that may be far apart in the protein sequence, but they are spatially collocated within the protein structure. So EPITOPE 3D is a tool that uses machine learning model combined with structure signatures to predict conformational epitopes. So in this way, trying to identify epitopes could lead to a higher disease response in the design of vaccines and also in the immunotherapy process. And those two are our key motivation in this work. Before I share the step-by-step methods used to build EPITOPE 3D, I would like to reinforce some limitations we have in the conformational epitope prediction field. So based on our literature review, we were able to verify that most existing predictors have limited classification performance. Even those with greater accuracy value, but they present low F1 score and also Matthews correlation coefficient, or they are only using the area under the ROC curve as a metric to evaluate their performance. Another limitation important to highlight here is about the generalization. And we can see that when the training and the testing performance have a big gap between them. What does this mean? It means that the models aren't generalizing well to a new data set, or they are not showing the reality. And the last point is about a benchmark data set. Those used by previous approaches were either no longer available now or were used during their method evaluation, which make them inappropriate for us or for an independent and a fair comparison now. And these limitations are in part due to the computational challenge we have in this field. So the first, the selection of a plentiful epitope data to train a model, to train a machine learning model is one of them, since epitope is context dependent. And what's that mean? It means that a single antigen may present different epitopes around it. In each antibody interaction, it will present one different one. One more aspect is that we are dealing with a huge imbalance data points, because in general, epitopes represent only 4% of the antigen structure. And to complete, there is still no fixed characteristic or feature that could distinguish epitopes from the remaining antigen surface. With that in mind, we can move to the development. So in our development workflow, we have three main stages: the curation of the data, the feature extraction and selection, and the machine learning process. The first one is the data curation. Here we aim to identify unbound antigen structures, but based on experimental antibody-antigen complexes, and they were all extracted from the protein data bank. And the central idea of this approach is to reduce the false negative annotation. So different epitope regions are first aggregated, are first extracted, and then they are aggregated in one antigen based on sequence and also structure similarity. This step resulted in the larger non-redundant set today. It almost 3 times bigger than the previous report, achieving 245 antigen structures. And from this total set, we are releasing 45 structures as an independent set to evaluate the performance of other available methods as well. And at the end of this stage becomes more clear to us how imbalanced the data is. We can see here that less than 4% of the antigen structure are represented by epitopes, which imposes an extreme hardship for a machine learning scheme. The second stage is the feature engineering. We use the curated data to extract characteristics of both epitope and non-epitope residues. And we evaluated and adapted a mathematical concept called graph-based signatures to represent the geometry and the chemical composition of the environment surrounding an epitope. And the main idea behind this technique involves modeling atoms as nodes and their interaction as edges, besides labeling these atoms based on their physical-chemical properties as well. So with this network representation, distance-based patterns between atoms are extracted as a cumulative distribution function. It's important to remember that this approach has been successfully employed in a range of other applications, including the development of methods to predict effects of mutation on protein structure, protein function, and also on antibody-antigen interfaces. Several other features were assessed as well here, including secondary structure, disorder regions, position-specific scoring matrix over evolutionary time, B-factor score, amino acid composition, amino acid index, and many others. But in order to reduce the model complexity, the noise, and also to optimize the predictive performance, an incremental stepwise greedy feature selection approach was performed here as well. And the third and final stage is the machine learning process. Here, different supervised learning algorithms were evaluated by regular in addition to the stratified mode of cross-validation in 5, 10, and also 10-fold. Besides that, this process includes qualitative data analysis of the selected feature, the previous step, and the training and also the model optimization. And the top performance was achieved with AdaBoost, or adaptive boosting. That is an iterative ensemble method. And we evaluated it 20 independent times using the 10-fold cross-validation approach. And to sum up, I would like to share the summary of our results. Regarding the imbalance issue that I mentioned previously, we have reached the best outcome combining two strategies only on the training data points. So we first randomly undersample the majority class that are the non-epitope residues. And then we use an oversampling technique called SMOTE that synthetically creates epitope data points, but based on the existing ones. So with both strategies combined, we could decrease the imbalance ratio originally from 1 to 28, that was in the raw data points, to 1 to 2. So this improves the learning process. And we can see here that with this classifier, it's remarkable that it achieved a good performance on the cross-validation with a Matthew correlation coefficient of 0.55. And taking the benchmark set as a blind test here, remember the benchmark data set composed of 45 new antigen structures. So we take it and we compare our model with other well-known B-cell epitope prediction tools like CIPA, Discotope, ELIPRO, BPPRED. And as we can see in all the columns here, EPITOPE 3D outperformed all of them, reaching MCC of 0.45 and F1 score of 0.36. And even in a highly imbalanced set, here we have 1 to 13 imbalance ratio. Just to highlight another point here is that the Matthews correlation coefficient was chosen as a reference because it's a more reliable statistic rate which produces a high score only if the prediction obtained good results in all of the 4 confusion matrix categories. And to end the presentation, I would like to inform that our predictor is already available as a friendly web server in which the user can upload the protein structure file or just inform its PDB code. And I brought here an example of the results section showing the list of residues, the residue identifier, the residue name, the chain, and also the PDB position of this residue in the PDB structure file. And also this list shows the classification. Here we present the results as epitope or non-epitope. We also show the prediction probability score behind this classification. And there are other useful information in the web server. As example here, the 3-dimensional visualization of the whole protein, where the user is able to select one or multiple residues. It's also possible to color the entire structure or color based on epitopes and some other possibilities. So that's my presentation for today. Thank you all for the attention.